So hello, my name is Sebastian Springer and I'm a professor of biochemistry and cell biology here at the Jacobs University. I've been at Jacobs for a very long time and I'm in fact really one of the dinosaurs. And so we've also had a laboratory here, my group and I, for a long time, and we have trained and worked with many undergraduate students. So I thought I would tell you a little bit about undergraduate research opportunities in my laboratory, but perhaps first describe it a little bit. So we're interested in the immune system, and this means in the way that the body defends itself against pathogens, attackers from the outside, bacteria or viruses. And during my PhD thesis a long time ago, I was involved in figuring out a mechanism by which the immune system can recognize cells that are infected with a virus or with bacteria also. And we have continued all the long time to work on this particular mechanism, especially some protein called an MHC class 1 molecule. And those of the students who have been to my immunology class, they will know about this already very well. Now, we are not an immunology laboratory in the medical sense. This means we don't have any patients. We don't even have experimental animals, we don't work with tissues, we don't actually investigate the immune response in people. But you could describe our approach and our laboratory as a molecular immunology lab. This means we investigate these MHC class 1 proteins with the means of biochemistry. This means we produce them in bacteria or in some other way. We investigate them inside cells with how they traffic, where they go. So we use microscopy, for example. We use a technique called pulse labeling, or pulse chase experiments, immunoprecipitations, followed by SDS page, gel electrophoresis, maybe some Western blotting, something like this. So these would be the cell biological things that we do. And then we also investigate the same molecules with the means of biophysics. This means, again, we produce these proteins and then we perform biophysical experiments. That's, for example, we look at the denaturation of the protein, we look what is the melting temperature of the protein depending on which peptide it has bound, or how quickly does it bind its peptide, which peptides does it bind, and so on. And some time ago, we also used to do computer simulations, and maybe in the future we will do this again. So it's biochemistry, biophysics, cell biology, mostly these methods. And we try always to bind these things together to give one story. So many of our good papers, of course, I would like to say that all of our papers are good. Many of our good papers, they combine several of these methods, and that can be quite demanding, but it's also very interesting and beautiful. So molecular immunology, and I like it very much because I'm an example that somebody can work on one single protein for a very long time, but we now really know very well about what we do, and it's very satisfying. One of the good things that I like about what we do is that we have a close connection to application. We have made several inventions that have become patented. Those inventions that were patented, we licensed out to companies that used them to make products. And a few years ago, we even set up our own company that was active for a few years, and we were selling proteins, special proteins that can be used to detect T cells that are directed against tumors, so-called MHC tetramers. And that company that got sold after a couple of years, but maybe, who knows, there will be another one. So we are actually also quite close to application, and I have to say that I really enjoy that a lot. So with respect to undergraduate students in the laboratory, it really is necessary for somebody who works with us, that they have a good theoretical and practical background in the life sciences. So this could be somebody who is, pretty strong already from high school, who's done a lot of biology and chemistry already, and then they could approach us perhaps in the first year. In that case, we will know each other already from the general biochemistry class, which I teach in the first semester, which I like very much. You could also, as an undergraduate student, you could also approach us in the second year, maybe after the immunology lecture, where much of what we do becomes much clearer and more obvious. And for an undergraduate student who would like to perform their bachelor thesis with us, I think it would be a very, very good idea if they let me know at the beginning of the fifth semester, so that during the fifth semester that particular student can already gain some practical experience in the laboratory. We cannot take an infinite number of people, and the reason for that is very simple. It is because everyone who is in the lab also needs to be supervised by somebody, and we have, I would say, a limited supervision capacity. That's the reason for that is that we only have a limited number of PhD students and postdocs, and so normally we limit the number of undergraduates to those people who are actually later on intending to work with us for their bachelor's thesis. Sometimes there are exceptions. We have sometimes special extra projects. For example, we make a movie, or we put a teaching script together, or we write a review article or so. And in these cases, sometimes people can come in additionally, they can work for us for a time, and then they can leave again. And this is then nothing to do with a bachelor thesis. But by and large, the number of students who work in our lab is relatively small. It's never been more than 6 at a time, but sometimes it's also just been 1 or 2 during the semester. It depends on how many we can accommodate. And for the people who work with us, typically what they do is they perform experiments under supervision to begin with, and after some time, maybe they get to do their own experiments, especially if we know that they can do things well and reproducibly. One thing that we really insist on that's very, very important is that people understand that the work that undergraduates in the lab do is for real science. So it's not done for entertainment, it's done for real science. And this is, of course, very beautiful and nice to know that you're actually working for a real scientific paper. But on the other hand, it can also be frustrating, because it means that sometimes you have to repeat something many times, or you have to optimize something if the gel doesn't look beautiful, or if there's too much protein on it or something like this. So it can also be a frustrating experience, but then this is exactly how research actually works, right? That's how research goes. So, and the methods that people use are the methods that all of us use in the laboratory that I mentioned before. They're actually not so different from the methods that are used in other laboratories, in the life sciences, at Jacobs. At the undergraduate level, many of the lab work details are actually pretty similar. So somebody might, perform a cloning process in my lab, like cutting open a plasmid, putting an insert in, ligating it, putting it back into the bacteria, something like this. But they might do this also in the lab of Claudia Briggs or Matthias Ulrich, or maybe even in the lab of Matthias Winterhalter or so. So at the undergrad research level, the labs look very similar to each other. But what is different, of course, is that what people say and do, the other people in the lab, and that can be very interesting. So my invitation would be for those students who are working with us in the lab, that they actually also intellectually try to take part in the life of the lab, that they come to our group meetings, that they ask a lot of questions, they try to figure out what is the lab about. And this might be even more useful than the actual handwork in the lab. And this extends actually also to people who are unable to come and work in the lab because they have no time, or for whatever other reason. We still welcome people to join our group meetings, for example, and they can witness or take part in scientific discussions about particular subjects. They can see how science works. So that is an open invitation, because we can, in the meetings, we can accommodate any number of people, and you will see how students, postdocs, whoever, introduce their research findings, and then they get discussed. This can also be very interesting. And if you're interested to know more, then one important place that I would like to point you towards is our website. And this you can go to our website here at Jacobs University. You can go to that website and then read however much you want. There are links to our original papers if you're interested in reading those, but there's also description of our research and who is in the lab and so on. You can contact people from the lab, for example. So if you're interested in working with us, you can write to me, but you could also, if you know somebody who's working with us, a postdoc or PhD student or so, from maybe the fact that they're an RA or so, then you can go up and talk to them. Everyone likes to talk about their research, right? And so do I. Thank you very much. Take care and all the best.